basically what we are focusing now on gene-based therapies, mainly RNA-based therapy using antisense oligonucleotides or microRNAs or siRNAs, as well as DNA-based therapy. And when I talk about DNA-based therapy, I refer to gene editing tools such as CRISPR-Cas9. So if we have genetic disorders that we know that, they are caused by only one single gene, in theory, this should be easy for us to target with the CRISPR-Cas9 or with a specific antisense oligonucleotide. Why is that? Because if there is a mutation in one single gene, you can go and design CRISPR-Cas9 with the guide RNA to go directly to that mutation in that particular gene and cut the mutation. Maybe you can replace it with the normal version of the gene, or maybe you don't mind and just create a frame shift mutations. And this way you will guarantee there is a permanent modification. And in this case, with the CRISPR-Cas9, you are correcting the mutation. And this is exactly what we do. But for us, in order to cover all the strategies, we actually are currently conducting parallel strategies. So we are doing CRISPR-Cas9 as well as antisense, which is targeting the messenger RNA. And in the past, and we will continue, we are targeting proteins. When we talk about proteins, we are designing and thinking of specific drugs to target the mutant protein. And this is what traditional drug is doing. So I will show you maybe a couple of slides of our main focus, mainly with the CRISPR-Cas9 and antisense. So let's start with the antisense. Basically, we just published 2 years ago in Molecular Therapy Nucleic Acid Journal, and a very good approach where we were able to silence and replace RNA therapeutic approach for OPMD. And this is oculopharyngeal muscular dystrophy, is an autosomal dominant disease caused by only one single gene. And there is a mutation in the BAP1. What we did, we designed antisense oligonucleotides, basically microRNA and ribozymes, to target the mutation in BAP1 and to clear this mutant messenger RNA as well as the mutant protein BAP1. At the same time, we had no option but to introduce optimized codon BAP1 in order to keep the normal BAP1 protein intact. So this idea, we are currently pursuing it in our transgenic mouse model. And here you see our goal is basically to construct one single vector where we have our microRNA, which are the antisense oligonucleotides, as well as the optimized codon BAP1, in order to get more data from preclinical work so we can translate it into patients. So we are currently doing this project. At the same time, we actually approaching the CRISPR-Cas9. In fact, I am writing my manuscript on using CRISPR-Cas9 on OPMD. This is the first study where we show that this idea of genetic editing can work on this disease, on oculopharyngeal muscular dystrophy. What I did, basically, I designed many guide RNA to target the mutation in exon 1 of this gene, BAP1. And I was happy to find at least 6 different guide RNA were able to cut the mutation. And here, as you see, this is kind of a diagram showing the special nicase, special CRISPR-Cas9, because we want to reduce the off targets. So we use nicase in order to create only single strand break surrounding the mutation. And the idea here was tested in cells such as human cells, muscle cells, as well as myoblast cells from patients. So this slide shows basically the sequence of the experiment where we designed the guide RNA with the CRISPR-Cas9. We had to transfect the construct into cells like HIC293, and then we had to isolate single cells through FACS, and we had to confirm our gene editing by T7 endonuclease, but also we had to use the Sanger sequencing. So here is my my slide showing the same idea of the CRISPR-Cas9 that was able to correct BAP1 mutation not only in cell line but also in a human patient myoblast, as you see. And here is the proof of the Sanger sequence. So our idea is to use the same tool, which is actually doing it. We are doing it right now in our mouse model. So we will actually apply this in near future in a transgenic OPMD model, because our ultimate goal is to get more data from preclinical before we will go and translate our treatment strategies into patients. So yes, we are working hard on both the approaches, the RNA as well as the CRISPR-Cas9. One important maybe a point I need to clarify, in addition to antisense oligonucleotides that target any spot or any place in the messenger RNA, we were actually recently collaborating with a group of computer scientists and bioinformatics in Concordia University, where they developed for us an evolutionary algorithm. So instead of only taking one antisense oligonucleotide at a time and design it manually and see the mismatch if it can target the mutant allele while leaving the wild type allele intact, we actually use this bright idea of evolutionary algorithm where we can design thousands and thousands of RNA molecules, antisense oligonucleotides, and then our goal or our task in the lab is to test the top candidates in our cells and to see if some of them they are really able to target the mutant allele while leaving the wild type allele intact. So far, I can share some preliminary data with my student. So far, we have good results. Here I'm showing data from two diseases, MGD, which is polyglutamine diseases, OPMD is polyalanine diseases. Both are caused by only one single mutation in one single gene. And as you see from this picture, in lane number 2, the expression of a mutant attacks CONy3, but in lane 4, with the use of this antisense oligonucleotide that has been generated from this algorithm, we were very happy to see a big reduction of the level. And if you look carefully at lane number 1 and lane number 3, you see same protein expression. I'm talking about in number 1 and number 3, I'm talking about the wild type. So here the idea is to design antisense oligonucleotide that target only the mutant allele while preserving the expression of the wild type. And the same story here in BAP1. So we are actually, right now, we are continuing testing more hundreds and hundreds of different molecules because we are interested to do similar to these studies that have been published from other parts from the world. But here, what they did, they are doing similar idea like us, targeting the repeat, because I'm talking about diseases like polyalanine or polyglutamine diseases are caused by one single gene, but the mutation, the type of mutation is the repeat, either alanine repeat or polyglutamine. But in this study, basically, they were able to actually design these siRNA with a nanoparticle and target the SAG, but they did it manually and they did it one at a time. Our idea is it will be great if we can have a software or if we have an algorithm that will generate for all of these diseases, and then the researchers can come and test hundreds or even maybe a few of them and find the correct ones. Another approach that we can do is to target the SNP. The SNP here, basically, you can plan to design your antisense oligonucleotide to target this single nucleotide polymorphism that is associated with the mutation. But here in Huntington disease, researchers, they were able to do it, but this idea, you cannot apply it for all diseases because, for example, in OPMD, the project I am working with, I couldn't find any single nucleotide polymorphism that is associated with the mutant BAP1. So this is a very neat idea, but you cannot, as we said here, or as the slide said, it is basically personalized. You cannot generalize it for all diseases. But our approach to target the repeat itself and to be selectively to the mutant, I think it will be a promising idea. So just to sum up, I will just stop here to show that other studies around the world, they used also CRISPR-Cas9 in different diseases like Huntington disease, Alzheimer's disease, and ALS. And here they used it, actually some of these studies, they used it in transgenic mice. So we are optimistic and we feel that this CRISPR-Cas9 or gene editing can be an effective tool for in the future, in the near future, but also for long-term future for many neurodegenerative diseases as well as neuromuscular diseases. I would just like to add maybe one or two slides here with the advances in CRISPR-Cas9 technologies such as base editing and the prime editing. Researchers, they are able now to restore and to change even one or two nucleotides, and we are optimistic with these advances. Our goal in our lab at MNI at McGill is to continue working on parallel strategies for treating neurodegenerative and neuromuscular diseases. We will continue to be updated with the technologies, but also we are interested and dedicated to understand the full impact of treatments and to understand not only how these treatments they work and they improve the phenotypes, but also will continue understanding the cellular and the molecular mechanisms of the disease, because then we can target the disease effectively. But if we are facing single genetic disorders, I think the best approach is to continue our strategy with the gene-based therapy, mainly RNA therapy and CRISPR-Cas9 therapy. In this, I would like to thank my boss, Dr. Guy Rouleau, who's the director of MNI at McGill, all the members in my lab, our collaborators, the funding agencies, and big thanks to the CONy conference and to VG's Neurology for doing this interview with me. Thank you.